Hey guys, so today we're just going to be talking about, again, cellular respiration and the mitochondria because I know the video I showed you before was a little fast-paced, and I just wanted to review everything with you again to make sure you have a good foundation. So this video is basically about the steps, more specifically of cellular respiration. Then we are going to identify the structure of the mitochondria and how that structure allows the mitochondria to produce more ATP. Before we go into all the steps of respiration, let's just have a little recap on ATP. So remember from our last video on energy, ATP is the energy of our cells. And the reason it's so important, and it's basically the energy source that every organism uses, is because it has all of this energy stored in that last phosphate bond. So there's a lot of potential energy stored in that bond right there. And when you break that bond through by adding water to it, then all this energy can be released. And just to highlight that importance of that bond right there, the triphosphate is the most important bond in ATP. That's where all the energy is stored. And when that bond is broken, that energy is released. So now we have a little bit of a refresher on what ATP is and how it's recharged and used as an energy source. Now we can talk about cellular respiration in more detail. So cellular respiration is the process that converts glucose, right here, so that's a sugar, into ATP, our energy source. And it does that through a variety of steps that we already know as glycolysis, the Krebs cycle, and electron transport chain. So here's just the general formula. So you have glucose, and that's what you're starting with. That's your reactant. And then you also have oxygen, another reactant. And the products of this reaction are carbon dioxide, so that's a byproduct, water, also another byproduct, and lots of ATP. There's about roughly 36 to 38 ATP made. The process really isn't as efficient as we'd like it to be, so that's why they kind of have a range in different textbooks depending on what you look at. And again, just to highlight it, these are the reactants, the input of the reaction, and these are the products, the output of the reaction. And as you already know, and I said just a few minutes ago, there's three major steps. So there's glycolysis, the Krebs cycle, and the electron transport chain. And they all occur at different parts of the cell. So we're going to talk about each of those steps in a little bit more detail now. So the first step of cellular respiration actually occurs outside the mitochondria in the cytoplasm, and that process is glycolysis. So now we're going to talk about glycolysis in a bit more detail. All the rest of the reactions happen in the mitochondria. So here's another diagram, and we are in glycolysis in the cytosol. So you already saw this video, and basically it just highlighted that glycolysis converts glucose into a molecule called pyruvate. And it does that using a bit of ATP. So in this reaction, you have glucose as your input, as well as some ATP, and you're going to get out pyruvate. And this reaction, they highlight that it does not require energy. So that means it's an anaerobic process. It doesn't require energy. So glycolysis can occur whether there's energy in the environment or not. It doesn't really matter. The video you watch also highlighted that it makes 2 ATP and 2 NADH. So you're starting with glucose, and you're getting a net gain of ATP and a net gain of 2 NADH molecules. And NADH are just these enzymes that kind of hold electrons, and they carry them all the way to the electron transport chain because that's where they're going to be used later. So they're really, really important. So here's just another model of glycolysis, a part of cellular respiration. So you're starting with glucose, you're using some ATP to get the reaction to start. Then you are creating some NADH molecules using NAD plus and phosphate. And then you are generating some ATP. And throughout this process, you're kind of getting some energy and getting it transferred and released. And at the end of this, you have 2 pyruvate molecules. You moved from a 6 carbon sugar to 2 3 carbon sugars in the form of pyruvate. So just to highlight the reactants, so we have some glucose, we have 2 ATP molecules, that's starting the reaction, 2 NAD+, so those are the coenzymes before they are added with a hydrogen. And then you have 2 phosphates. Again, those are going to be used to produce your ATP. And then you have 4 ADP. So that's going to be combined with the phosphate to create ATP, that recharged energy source. Then you have your products. So your products are what's coming out of the reaction. So you generate 4 ATP, but because you use 2 in the beginning, you have a net gain of 2 ATP. So you have 2 pyruvate, so that's the 2 pyruvate. The 6 carbon has turned into 2 3 carbons. That's why you have 2 pyruvate. You have 2 NADH molecules. That's coming from the NAD plus and the hydrogen. So they are making an NADH molecule, those coenzymes that are going to hold those electrons. So glycolysis, we've already talked about, is on the outside of the mitochondria, and that makes some ATP, but not a lot. So let's go and make some more ATP. So before we can start the Krebs cycle or the citric acid cycle, something else needs to happen. So pyruvate needs to be converted into this molecule called acetyl-CoA before anything else can start. So I don't really need you to know the details about this reaction, but just know that pyruvate, that is converted into acetyl-CoA right before the Krebs cycle. And after that process, you have some CO2 as a byproduct and also another NADH molecule. So this is really important. Before we can go into the Krebs cycle, we need to generate acetyl-CoA. And now finally, now that we've created our acetyl-CoA, we can actually enter the mitochondria and go through the Krebs cycle. So now we are in the mitochondria conducting the Krebs cycle. So let's talk a little bit more about the mitochondria and its structure. So the mitochondria allows for the production of more ATP based on its structure. So it has an outer membrane that's basically like the skin, and then it has this inner membrane. So that inner membrane has many folds called cristae. So all of these different folds, we talked about surface area before. This increases the surface area. So more folds allows more places for ATP production. So there's tons of folds in here that kind of increases the area that you can have more reactions occurring. And then in here, the matrix, there's tons of enzymes there ready for ATP production. So the mitochondria's folds are really important because it allows more reactions to occur. So basically, more space, more work can get done. And we already saw this picture, but now we are right here in this picture in the Krebs cycle or the citric acid cycle. Alright, so here's the citric acid cycle. Get to it. Memorize this reaction. No, I'm just kidding. We're not doing this reaction. This is way, way too detailed and so much chemistry oriented. It's crazy. So let's just give you the basics that you need to know. So you already saw this video, but the Krebs cycle mainly produces the majority of the NADH and FADH that will be used later to produce ATP. And it makes some ATP, but again, not a lot. So this process requires oxygen, so it's an aerobic process. So unlike glycolysis, this reaction needs oxygen to occur. No oxygen, no Krebs cycle. So you're taking acetyl-CoA, and that's going to be used to use more NADH and this other coenzyme called FADH2. So basically that does the same exact thing NADH does, it's just a different molecule. It holds electrons. So here's just another representation. You're starting with acetyl-CoA, you're breaking it up into different molecules, and along the way you're creating some CO2, some NADH, some more CO2, some more NADH, 2 ATP, 2 FADH, and 2 NADH. So your total products would be 2 ATP are gained, you make 6 NADH, 2 FADH, So these are both electron carriers. So you're making a lot of electron carriers in this reaction. And 2 CO2. I'm sorry, actually 4 CO2. Don't know how I missed that, but it's okay. So the last and final step is the electron transport chain. And this is where all that ATP is actually going to be produced. Right now, we've only made 4 ATP. That's not a lot. And once again, the electron transport chain is in the mitochondria still. So we're still in that mitochondria. So the electron transport chain is basically like transporting of electrons. So and these electrons are holding energy. So you're kind of transferring the energy. It's like going down steps. You're transferring from high to low. And eventually you're going to have that formation of water and the formation of ATP. So I'm going to try to keep this brief. But the first thing that happens, there's basically multiple proteins embedded within this mitochondria membrane. So the first type of protein and enzyme basically takes the electrons from NADH and transfers them through the protein throughout the membrane of the mitochondria. And in the process, hydrogen protons are released and put into that intermembrane space, creating that gradient that we mentioned before. And there's another protein, same for FADH2. So that will take the electrons from FADH2 and transfer them along this gradient. And then throughout this entire process, hydrogen ions are being moved from a high concentration, so out in the membrane space, to a low concentration in the membrane space. And then those electrons are basically carried throughout. And then this proton gradient basically drives this ATP synthase. So it's basically the potential energy from that gradient is driving this protein, the ATP synthase. And that ATP synthase is what's going to produce your ATP. It's going to have ADP and phosphate within here. And then this protein will basically attach those phosphates together, creating ATP. So key points here is that diffusion causes hydrogen protons to flow into the inner membrane space of the mitochondria. That protein gradient has potential energy, and that's what's driving the ATP synthase to create ATP. Energy is transferred from those carrier electrons. That's basically driving the reaction again. And if you want more details specifically on this process and about each of the proteins, you can come and see this link right here. And finally, the last thing that's most important is that oxygen is basically that final electron acceptor. So we know that water is made in this reaction. Basically, water is made from it accepting those hydrogen ions at the end. So at the end of the reaction, it will accept hydrogen ions and it will produce this water as a byproduct in this reaction. It's the final electron acceptor. So that's where you're getting water from. Oxygen that's kind of floating inside the mitochondria will bind with the hydrogen protons that were left over from the gradient and it will produce water. Just to go back to the original formula we started with. So in this formula we started with glucose and we used oxygen during the Krebs cycle and the electron transport chain, and we're left with carbon dioxide both from the Krebs cycle and we have water from the electron transport chain, and we're mainly producing all of this ATP. So the key points of this presentation is that mitochondria's structure allows for the production of more ATP. More surface area means more space for proteins that can help generate ATP. Respiration has many steps, but the main reason is to produce this ATP. And the final product of respiration, again, is many, many ATP molecules. So I hope this anything that you were confused about and had questions about. We'll talk more about this stuff in class, but I hope you learned something. Bye, guys.